So here's something worth thinking about as you settle in tonight. Right now, as you lie here, your kidneys are working. Not resting, not slowing down, not waiting for morning. Working. At this exact moment, roughly 1.5 liters of blood per minute is flowing through two small organs sitting just below your rib cage, one on each side of your spine, each about the size of your fist. That blood enters through enormous vessels, passes through one of the most intricate filtering structures in all of biology, and exits, cleaner, more precisely balanced, carrying exactly the right concentration of salts and minerals and water to keep everything in your body functioning the way it's supposed to. And the filtered waste, the urea, the creatinine, the excess potassium, the metabolic byproducts of everything you've eaten and thought and done today, is being concentrated into urine and routed quietly toward your bladder. You will not feel any of this happening. You don't need to. Your kidneys will do it without you. By the time you wake up tomorrow morning, your kidneys will have filtered your entire blood supply something like 60 times over. In a single day, they process about 180 liters of fluid. Not 180 liters of blood, but 180 liters of filtrate, the raw material they squeeze out of your blood, and then, with extraordinary precision, sort through. Of that 180 liters, they'll return about 178.5 liters back to your body, keeping only the waste. About 1.5 to 2 liters becomes urine. The rest, water, glucose, amino acids, vitamins, the minerals you need, gets reclaimed, recovered, sent back. Nothing is wasted. Nothing gets through that shouldn't. The kidneys maintain, with a precision that no hospital instrument can fully replicate, the exact chemical environment that every cell in your body requires to live. And almost nobody thinks about this. People think about their hearts, which they can feel beating. People think about their lungs, which they can feel breathing. But the kidneys work in silence, in the dark, without feedback, without sensation, usually without any acknowledgement at all, until something goes wrong. And when something goes wrong with your kidneys, you discover very quickly how much you had been taking for granted. That's what we're going to talk about tonight. Your kidneys. Literally everything about how they work, from the structure of the individual cells that do the filtering, to the hormones they produce, to the remarkable history of how we figured any of this out, to what happens when the system breaks down, to the people who invented machines to replace them when they did. We're going to take it slow, start from the beginning, walk through it together, no rush. If you drift off somewhere along the way, that's fine. That's the point, actually. These ideas will still be here. And honestly, there's something particularly right about learning how your kidneys work while they're working. Quietly, competently, doing what they always do, the same tonight as every other night of your life. You don't need to know any biology to follow this. You really don't. Because the kidney at its heart is solving a problem you already understand intuitively. How do you keep a system in balance? How do you take in everything from the outside world? All the salt, all the water, all the protein, all the minerals, and maintain, despite all that variation, a stable internal environment. That's the kidney's job. Balance, homeostasis, the ancient Greek idea that the body, to function, must hold itself in a kind of dynamic equilibrium, not static, not frozen, but constantly adjusting, constantly correcting, always returning to center. The kidney is the organ most responsible for that return. We're going to take our time, and we're going to start not with anatomy or chemistry, but with a question that puzzled physicians for thousands of years. What, exactly, is urine? Where does it come from? Why does it exist? The answer, it turns out, is not obvious at all, and the history of how people got it wrong, and then slowly got it right, is one of the more quietly interesting stories in all of medicine. Let's start where the story actually starts, which is not in a kidney, but in a centuries-long argument about what urine is and where it comes from. For most of human history, the answer to that question was the liver. This is what Galen thought. Galen of Pergamon, the Greek physician who lived from around 109 to 216 CE, and whose ideas about medicine dominated Western thought for well over 1,000 years. Galen believed that urine was a kind of byproduct of blood formation. The liver, in Galen's system, was the central organ of the body, the place where food was transformed into blood. And urine, he thought, was the residue of that process, a watery waste product that drained somehow from the liver through the kidneys and into the bladder. The kidneys, in this view, were little more than a drainage pipe, a passive channel. They didn't do anything interesting, they just let the urine pass through. This was wrong, profoundly, completely wrong. But it persisted for an astonishing length of time because nobody had a good way to test it, and because Galen's authority was so total in medieval European and Islamic medicine that challenging him on any point was an intellectual and sometimes physical risk. You studied Galen. You commented on Galen. You did not contradict Galen. The first serious cracks in the Galenic view of the kidneys appeared in the 16th century, during the great explosion of anatomical investigation that came with the Renaissance and the rediscovery of direct observation as a method. Andreas Vesalius, the Flemish anatomist who published his landmark work De Humani Corporis Fabrica in 1543, was one of the first to insist that anatomy should be done by cutting open actual bodies and seeing what was there, not by reading what Galen said should be there. Vesalius corrected dozens of Galenic errors through direct observation, and while he didn't fully solve the kidney problem, he began to shift the intellectual culture toward one where solving it was possible. The real breakthrough came in the 17th century, with the discovery of something that seems obvious now, but was genuinely revolutionary at the time, that the kidneys actively make urine, not passively drain it, make it, transform it. The blood that enters the kidney is different from the blood that leaves it, and the kidney is responsible for that difference. This insight came from a variety of investigators working in different places, from Lorenzo Bellini in Florence, who in 1662 described the tubular ducts in the kidney through which urine passes and argued they were involved in filtration, from Marcello Malpighi, the brilliant Italian microscopist who in the 1660s used the newly invented microscope to examine kidney tissue and discovered the tiny spherical structures, the glomeruli, where filtration begins, from a growing body of experimental work showing that animals whose kidneys were removed quickly died in a distinctive way, accumulating waste products in their blood, a condition we now call uremia. Malpighi's discovery of the glomeruli was particularly important, though it took another 2 centuries to fully understand what he'd found. Under his microscope, the kidney revealed itself as something far more intricate than a simple drain. There were these tiny spheres, densely packed, each one surrounded by a tangle of capillaries, and there were tubes, long, winding, convoluted tubes, extending from the spheres through the kidney tissue, and there was a pattern to all of it, a structure, something that looked less like a passive channel and more like an active machine. Just sit with that for a second. Malpighi, peering through his primitive microscope in 17th century Bologna, was looking at the nephron, the functional unit of the kidney, without knowing what it was, without knowing what it did, just seeing that it was complicated, that it was organized, that there was more going on here than anyone had imagined. It would take until the 19th century before someone could explain it. In the interval between Malpighi's observations and Bowman's explanation, a great deal of productive confusion accumulated around the kidney. Various investigators proposed various theories. Some thought the kidney filtered blood through simple pressure, like squeezing juice from a sponge. Others thought it secreted urine actively, the way glands secrete their products, that the tubule cells were not passive conduits, but active producers, manufacturing urine from components of the blood. This debate, filtration versus secretion, occupied renal physiologists for more than 2 centuries. Both sides were partly right, which made the debate particularly difficult to resolve. The filtration side was championed most influentially by the German physiologist Carl Ludwig, who in 1844 proposed what he called the filtration reabsorption theory, that the glomerulus produces a filtrate by simple physical pressure, and that the tubule then selectively reabsorbs what the body needs. This is, in its essentials, correct. But Ludwig couldn't prove it at the time, because the tools to measure what was in the filtrate compared to what was in the blood didn't yet exist. The secretion side, championed by Rudolf Heidenhain among others, argued that the tubule cells were actively secreting specific molecules into the urine, as well as reabsorbing others. This is also partly correct. The tubule does actively secrete certain molecules, including some drugs and toxins, into the lumen, but it was wrong as a primary mechanism of urine formation. The resolution came in the 1920s and 1930s, largely through the work of an American physician and physiologist named Alfred Newton Richards. Richards developed a technique called micropuncture, inserting a tiny glass pipette drawn to a point just a few micrometers across, directly into the tubule of an anesthetized frog or salamander, and withdrawing tiny samples of fluid for chemical analysis. For the first time, someone could directly measure the composition of the fluid at different points along the nephron, and Richards found what Ludwig had predicted. The composition at the start, in Bowman's capsule, was essentially identical to blood plasma filtered of proteins. The tubule was indeed taking a filtrate and then selectively recovering components from it. The filtration reabsorption theory was correct. Ludwig had been right for 80 years without proof. Richards provided the proof, and the basic framework of how the kidney works snapped into its modern form. This history matters. It's a reminder that some of the most fundamental things about how your body works were genuinely unknown, genuinely controversial, genuinely contested among serious scientists within the 20th century. The loop of Henle's function wasn't fully understood until the 1950s and 1960s. The molecular identity of aquaporins wasn't known until 1992. The circadian regulation of kidney function is still being actively worked out. The organ that has been keeping you alive every minute of your life is still, in some of its workings, an active subject of research. We know a great deal. We don't know everything. That someone was a British physician and anatomist named William Bowman. Bowman was born in 1816 in Nantwich, a market town in Cheshire, and he was, by all accounts, one of those rare people who combine extraordinary technical skill with genuine intellectual curiosity in roughly equal measure. He trained as a surgeon, but what he really loved was looking at tissues under the microscope, examining the fine structure of biological materials, trying to understand how organs did what they did by understanding what they were made of. And in 1842, when he was 26 years old, he published a paper on the structure of the kidney that changed everything. Bowman described, in precise detail, the connection between the glomerulus, Malpighi's tiny sphere of capillaries, and the tubule that extended from it. He showed that each glomerulus was enclosed in a cup-shaped membrane, like a hand cupped around a ball, and that this cup was continuous with the beginning of the tubule. The cup collected fluid filtered from the blood in the glomerular capillaries and channeled it directly into the tube. This cup, inevitably, came to be called Bowman's capsule, and the entire structure, the glomerulus plus Bowman's capsule, plus the long winding tube that followed, was the nephron. Bowman's paper didn't fully explain how filtration worked, or why the kidney could recover so much of what it filtered, but it identified the architecture. It showed that the kidney wasn't just a series of tubes, but a collection of millions of tiny independent filtering units, each one a complete system in miniature, each one performing the same operation, taking blood in, filtering out fluid and small molecules, and then processing that filtrate through a long tube that would selectively recover what the body needed and let the waste go. And now that we have the history, let's actually talk about what those units do, because understanding the nephron, really understanding it, down to the level of what happens at each stage, is understanding the kidney. Everything else is elaboration. Each of your kidneys contains roughly 1 million nephrons, 1 million tiny filtration units working in parallel, doing the same thing simultaneously. When you add up both kidneys, that's about 2 million nephrons, and each one is doing its job right now, tonight, as you breathe slowly and your body begins to settle towards sleep. The nephron begins at the glomerulus. The glomerulus is a tiny knot of capillaries, blood vessels so fine that red blood cells must pass through them in single file, pressing against the vessel walls. These capillaries are not ordinary capillaries. Their walls are riddled with tiny holes, called fenestrations, windows, essentially, pores in the capillary wall through which fluid can escape. The pressure of the blood flowing through these capillaries forces fluid outward, through the fenestrations, across a filtering membrane, and into the space inside Bowman's capsule. But the glomerular filter is more complex than just a holy wall. Outside the capillary endothelium, the layer of cells that lines the inside of the blood vessel, there is a membrane called the glomerular basement membrane, a thin sheet of specialized proteins that acts as a second filter. And outside that membrane, clinging to the capillary surface like a collection of octopuses, are cells called podocytes. Podocytes are among the most structurally extraordinary cells in the body. Each one extends a series of long projections called primary processes, and from those extend even finer projections called foot processes, pedicels, that interdigitate with the foot processes of neighboring podocytes. The spaces between these interdigitating foot processes are called filtration slits, and they are bridged by a thin membrane, the slit diaphragm, which is essentially the final filter. Molecules trying to get from blood into the filtrate must pass the endothelial fenestrations, then the basement membrane, then the filtration slits. Things that are too large, proteins, red blood cells, anything with a molecular weight above about 65,000 Daltons, don't make it through. The result of all this filtering is the glomerular filtrate, essentially blood plasma, minus the large proteins and cells. Water, salts, glucose, amino acids, urea, creatinine, small vitamins, ions, all of it passes through. In a healthy person, about 125 milliliters of this filtrate forms every minute. That's the glomerular filtration rate, the GFR, a number that nephrologists use constantly as a measure of kidney function. Over 24 hours, that's about 180 liters of filtrate. The fluid portion of your entire blood volume is cycled through this process many times over each day. Now here is where the kidney earns its reputation for complexity, because most of that filtrate, almost all of it, gets reabsorbed. If you simply let 180 liters of filtrate go, you would lose all your blood volume in about 20 minutes and die. The filtrate is raw material, not waste. It's the starting point for a process of sorting that happens in the tubule. the long winding pipe that follows Bowman's capsule, and that, segment by segment, recovers what should be recovered and lets go of what should be let go. The tubule has 4 distinct sections, and each one does something different. The first is the proximal convoluted tubule, which begins right at Bowman's capsule and is called convoluted because it winds back and forth, dramatically increasing its surface area for absorption. The cells lining the proximal tubule are packed with mitochondria. They're metabolically intense cells doing a lot of active work, and their inner surface facing the lumen of the tubule is covered with microvilli, tiny finger-like projections that also increase surface area. This section of the tubule is where the heavy lifting of reabsorption happens. In the proximal tubule, about 65% of the filtered water is reabsorbed, along with virtually all of the filtered glucose and amino acids, most of the filtered sodium, potassium, chloride, bicarbonate, calcium, phosphate, essentially everything the body values, pulled back out of the filtrate and returned to the blood. The glucose reabsorption is particularly impressive. Normally, the proximal tubule recovers every last molecule of glucose from the filtrate, which is why healthy urine contains essentially no sugar. When blood glucose rises high enough, as in uncontrolled diabetes, the proximal tubule's transport capacity is overwhelmed and glucose appears in the urine. That's glucosuria, one of the classic signs of diabetes, and it was observable for millennia before anyone understood what it meant. Ancient physicians actually tasted urine as a diagnostic tool. Mellitus, in diabetes mellitus, means honey sweet. The kidney's failure to reclaim glucose was visible long before the kidney's mechanism for doing so was understood. From the proximal tubule, the filtrate enters the loop of Henle, and the loop of Henle is where the kidney does something genuinely strange, something that, once you understand it, seems almost impossibly clever for a structure that evolved without anyone designing it. The loop of Henle is named after Friedrich Gustav Jakob Henle, a German anatomist who was, by all accounts, one of the most energetic scientific minds of the 19th century. Henle was born in 1809 in Fürth, in Bavaria, and he had the kind of broad intellect that was more possible in the 19th century than it is now. He made important contributions to anatomy, histology, pathology, and cell theory, and he was an early proponent of the germ theory of disease, arguing in 1840 that infectious diseases were caused by living organisms, years before Koch and Pasteur provided the experimental proof. He was Robert Koch's mentor, as it happens. The loop of Henle, which he described in 1862, is perhaps his most lasting legacy, even though he didn't fully understand what it did. He described the structure. The function took another century to work out. The loop of Henle is shaped exactly as its name suggests, a hairpin loop. The filtrate descends from the cortex of the kidney, the outer layer, deep into the medulla, the inner layer, and then turns around and ascends back to the cortex. The descending and ascending limbs of the loop are doing different things, and the interplay between them generates a concentration gradient in the kidney tissue that makes everything else the kidney does possible. Here's how it works. The descending limb of the loop is highly permeable to water, but not to salt. As the filtrate descends deeper into the medulla, water leaves the tubule by osmosis, drawn out by the increasingly salty environment of the surrounding tissue. The filtrate becomes more and more concentrated as it descends. Then it turns the corner. The ascending limb is the opposite. It's impermeable to water, but actively pumps salt, specifically sodium and chloride, out of the filtrate and into the surrounding tissue. As the filtrate ascends, it loses salt but can't lose water, because the ascending limb won't let water through. The filtrate becomes more and more dilute as it rises. And here's the beautiful part. The salt being pumped out of the ascending limb is pumped into the tissue around the descending limb, which is the same tissue the descending limb is sitting in. So the salt concentrates the medullary tissue, which draws more water out of the descending filtrate, which increases the concentration of the descending filtrate, which generates a bigger gradient to drive salt out of the ascending limb. The two limbs are counteracting each other in a way that amplifies the concentration gradient. At the top of the loop, near the cortex, the tissue is close to blood plasma concentration, about 300 milliosmoles per kilogram. Deep in the inner medulla, the tissue concentration can reach 1200 milliosmoles per kilogram, 4 times as concentrated. The kidney builds this gradient and uses it. The gradient is used in the collecting duct, the final section of the tubule, which passes back down through the medulla on its way to the renal pelvis. The collecting duct is where the kidney makes its final decision about how much water to put in the urine. If the body is dehydrated and needs to conserve water, a hormone called antidiuretic hormone, ADH, also called vasopressin, is released from the pituitary gland at the base of the brain. ADH makes the collecting duct permeable to water. As the collecting duct descends through the increasingly salty medulla, water is drawn out of it by osmosis. Concentrated urine remains, and water is recovered. If the body is well hydrated and doesn't need to conserve water, ADH levels fall. The collecting duct becomes impermeable to water, and the dilute filtrate flowing through it passes out as dilute urine. Your kidneys can produce urine ranging from about 50 milliosmoles per kilogram, very dilute like water, to about 1,200 milliosmoles per kilogram, very concentrated, dark yellow, the kind you produce first thing in the morning after a night of not drinking. That range, by the way, is extraordinary. The kidney can vary the concentration of urine by a factor of 24. No artificial filtering system comes close. Between the loop of Henle and the collecting duct is the distal convoluted tubule, the last section of fine-tuning before the filtrate enters the collecting duct. The distal tubule is where the kidney does the final adjustments to sodium, potassium, calcium, and acid-base balance, and where several important hormones exert their effects. One of the most important is aldosterone. Aldosterone is a steroid hormone produced by the adrenal glands, the little triangular glands that sit on top of each kidney, though they're actually part of a completely different system. Aldosterone acts on cells in the distal tubule and collecting duct to increase sodium reabsorption and potassium excretion. More sodium in the blood means more water follows it osmotically, which means more blood volume, which means higher blood pressure. Aldosterone, in other words, is one of the primary long-term regulators of blood pressure in the body. And the kidney doesn't just respond to aldosterone, it helps produce it. This is where the story gets particularly interesting, because the kidney is not just a filter, it is also an endocrine organ, a gland that secretes hormones, and the hormones it produces ripple through the body in ways that most people don't associate with kidneys at all. The most elaborate of these hormonal systems involving the kidney is the renin-angiotensin-aldosterone system, sometimes called the RAS, and it is one of the most elegant feedback loops in all of physiology. Here's how it works. Sitting in the wall of the afferent arteriole, the tiny artery that brings blood into the glomerulus, are specialized cells called juxtaglomerular cells. These cells are constantly monitoring the pressure and volume of blood flowing into the kidney. When blood pressure drops, or when blood volume falls, or when sodium concentration in the tubule at that point is too low, the juxtaglomerular cells respond by releasing an enzyme called renin into the bloodstream. Renin then acts on a protein called angiotensinogen, which is produced by the liver and circulates in the blood constantly. Renin cleaves angiotensinogen into a smaller molecule called angiotensin 1. Angiotensin 1, in turn, is cleaved by another enzyme, angiotensin-converting enzyme, or ACE, which is found primarily in the lungs, into angiotensin 2. Angiotensin 2 is a powerful molecule. It does several things simultaneously. It constricts blood vessels throughout the body, immediately raising blood pressure. It acts on the brain to stimulate thirst, prompting you to drink more water. It acts on the pituitary to increase ADH release, causing the kidneys to retain more water. And it acts on the adrenal glands to stimulate the production and release of aldosterone, which then acts on the kidney's tubules to increase sodium reabsorption. More sodium, more water following the sodium, more blood volume, higher blood pressure. The feedback loop closes. This system, renin, angiotensin, aldosterone, feedback, is also why sodium intake is so intimately tied to blood pressure. A diet high in sodium chronically expands blood volume, raises the set point around which the renin-angiotensin-aldosterone system operates, and over years, in susceptible individuals, produces sustained hypertension. The kidney is trying to defend a sodium balance, but the defense mechanism involves maintaining a blood pressure that is too high for the arterial walls to bear indefinitely without damage. Reducing sodium intake, a public health message that has been issued repeatedly for 50 years, with partial success, works directly through the kidney. Less sodium in the body means less volume to maintain, means less pressure needed to sustain that volume, means lower blood pressure. Every millimeter of mercury reduction in systolic blood pressure translates into reduced risk of stroke, heart attack, and kidney disease. The arithmetic is unambiguous. The kidney is at the center of it. Hypertension affects roughly 1 in 3 adults in the United States, and estimates suggest that inadequate kidney response to sodium excess, a genetic and environmental variation in how tightly different people's kidneys cling to sodium, explains a significant fraction of that burden. Some people's kidneys are salt-sensitive. They retain sodium more readily when intake is high and release it more reluctantly when intake is low. These people develop hypertension more readily on high-salt diets. Others are salt-resistant. Their kidneys adjust sodium excretion efficiently, and their blood pressure remains stable across a wide range of sodium intake. The genetic underpinning of salt sensitivity is complex, involving variants in genes encoding multiple components of the renin-angiotensin-aldosterone system, sodium transporters in the tubule, and aldosterone signaling pathways. It's an active area of research, and it holds the promise that someday we might be able to identify salt-sensitive individuals early and give them targeted advice. Reduce sodium before hypertension develops, rather than waiting for the damage to accumulate. This is why ACE inhibitors, drugs that block the enzyme that converts angiotensin 1 to angiotensin 2, are among the most widely prescribed medications in the world. By blocking that conversion, they interrupt the cascade. Less angiotensin 2 means less vasoconstriction, less aldosterone, less sodium retention, lower blood volume, lower blood pressure. The kidney's hormone system, when it's working too hard, can be one of the drivers of chronic hypertension. Blocking part of the system brings pressure down. Just sit with the elegance of this for a moment. The kidney is sensing its own blood supply, and when that supply drops, it releases a signal that travels through the blood, acts on another organ, produces a hormone, comes back to the kidney, and tells the kidney to hold on to more sodium and water, which raises blood pressure, which restores the kidney's blood supply. A closed loop operating continuously, keeping pressure in range. And if that loop is set a little too high, if the juxtaglomerular cells are a little too sensitive, or if angiotensin II is a little too active, you get hypertension, and the treatment is to reach into the loop and turn one of its knobs down. But that's not all the kidney makes, not by a long way. In 1906, a physiologist named Paul Carnot injected plasma from anemic rabbits into healthy rabbits and found that the healthy rabbits started producing more red blood cells. Something in the plasma of the anemic animals was signaling the bone marrow to work harder. He named this putative factor hemopoietin. He was right about the signal, but wrong about where it came from. It took decades to work out that the signal, the hormone, was being produced not by the blood, but by the kidneys. That hormone is erythropoietin, usually called EPO. When oxygen levels in the blood fall, as happens in anemia or at high altitude, or whenever the blood is carrying less oxygen than it should, cells in the kidney detect the drop and respond by secreting EPO into the bloodstream. EPO travels to the bone marrow and binds to receptors on erythroid progenitor cells, the cells that will become red blood cells, stimulating them to proliferate and mature. More red blood cells means more capacity to carry oxygen. The feedback loop closes. Oxygen levels recover. This is why kidney disease leads to anemia. When kidney tissue is damaged or destroyed, the cells that produce EPO are lost. EPO levels fall. The bone marrow doesn't get the signal it needs. Red blood cell production drops, and the patient becomes chronically anemic, tired, pale, short of breath, even when their lungs and heart are fine. The anemia of chronic kidney disease was mysterious for a long time, attributed vaguely to bone marrow suppression, until the EPO connection was understood in the 1970s and 1980s. And EPO, once identified, became one of the most important therapeutic discoveries in medicine, and also one of the most notorious. Synthetic EPO, produced by recombinant DNA technology, became available in the late 1980s and transformed the treatment of anemia in kidney disease patients, dialysis patients, and cancer patients undergoing chemotherapy. It also became, infamously, the doping agent of choice in endurance sports through the 1990s and 2000s, because artificially raising EPO levels causes the bone marrow to produce extra red blood cells, which carry more oxygen to muscles, which dramatically improves aerobic performance. The Tour de France scandal, Lance Armstrong, the systematic doping programs in cycling and distance running. EPO is at the center of all of it. A molecule your kidneys are producing right now, as you lie here, calibrated to the oxygen content of your blood, became one of the most powerful performance-enhancing drugs ever discovered. Your kidneys are, among other things, a natural EPO factory. The third major hormone produced by the kidneys, though produced is a slight simplification, is the active form of vitamin D. You may know vitamin D as the sunshine vitamin, the thing that forms in your skin when you're exposed to ultraviolet light. But what forms in your skin is not the active form. What forms is a precursor, vitamin D3, cholecalciferol, which is then processed by the liver into another precursor, 25-hydroxyvitamin D, which is then converted by the kidneys into the active hormone, calcitriol, also called 1,25-dihydroxyvitamin D. Calcitriol is what does the work. It acts on the intestine to increase absorption of calcium from food. It acts on the bones to regulate calcium deposition. It acts on the kidney itself to reduce calcium excretion in urine. It's a fundamental regulator of calcium metabolism throughout the body, and without it, calcium absorption from food drops dramatically. Calcium is leached from bones, and bone disease, rickets in children, osteomalacia in adults, develops. When the kidneys fail, calcitriol production falls. Calcium absorption from food drops. Calcium is pulled from bones to maintain blood calcium levels. Patients with chronic kidney disease develop a characteristic bone disease, renal osteodystrophy, and this was one of the puzzles of dialysis in its early years. Patients on dialysis were surviving, but their bones were deteriorating. It took time to understand that the kidneys were producing something the bones needed, and that dialysis, for all its ability to filter waste, couldn't replace that function. Today, dialysis patients take synthetic calcitriol as a supplement, but it's a reminder that the kidney is far more than a filter. Let's talk about acid-base balance, because it's one of the kidney's most important jobs and one of the least appreciated. The pH of your blood must be maintained within an extraordinarily narrow range, between 7.35 and 7.45. That's it. Slightly below 7.35 is acidosis. Slightly above 7.45 is alkalosis. Both are dangerous. An arterial blood pH below 7.2 or above 7.6 is potentially life-threatening. And yet your body is constantly generating acid from cellular metabolism, from the carbon dioxide produced by every cell that combines with water to form carbonic acid, from the breakdown of proteins into amino acids that carry acidic side chains, from the production of lactic acid during exercise, from a dozen other sources. The body has multiple systems for handling this acid load, and the kidneys are among the most powerful. Not the fastest, but the most durable. The lungs handle acid-base balance quickly. When blood becomes more acidic, you breathe faster, blowing off carbon dioxide, which shifts the equilibrium away from carbonic acid and toward lower acid levels. This happens in seconds to minutes, but the lungs can only deal with the carbon dioxide part of the acid load. They can't excrete fixed acids. Sulfuric acid, phosphoric acid, the products of protein metabolism. That's the kidney's job. The kidney handles acid-base balance by adjusting how much bicarbonate it retains and how much hydrogen ion it excretes. Bicarbonate is the blood's main chemical buffer, a molecule that can absorb hydrogen ions, neutralizing them. When blood becomes more acidic, the kidney increases bicarbonate reabsorption and increases hydrogen ion secretion into the urine. Urine can be made quite acidic, down to about pH 4.5, which is about as acidic as tomato juice, as the kidney loads it with excess hydrogen ions. When blood becomes more alkaline, the kidney reverses the process. Less bicarbonate is reabsorbed, more is excreted, and urine pH rises. The kidney can't respond as fast as the lungs, but it can sustain its response indefinitely and handle acid loads that would overwhelm the respiratory system. This is why chronic lung disease often leads to acid-base abnormalities, and why those abnormalities can be partially compensated by the kidneys. And it's why kidney failure so often leads to metabolic acidosis. When the kidneys can't excrete enough acid or retain enough bicarbonate, the blood gradually becomes more acidic, with effects that range from fatigue and confusion to serious cardiac arrhythmias. Now, let's talk about what urine actually is, because most people have a vague impression of it as a kind of liquid waste without much thought about what exactly is in it. Urine is about 95% water. Of the remaining 5%, the largest component is urea, a molecule made in the liver when proteins and amino acids are broken down. The nitrogen in those amino acids has to go somewhere. In fish, it can simply be excreted as ammonia, which is highly toxic but easily diluted in surrounding water. Land animals can't do that. They don't have unlimited water to dilute ammonia in, so the liver combines two molecules of ammonia with carbon dioxide to form urea, a far less toxic molecule, which the kidneys filter out and excrete. Urea is the primary carrier of nitrogen waste in mammals. Every protein you eat, Every muscle protein that turns over, every metabolic process involving amino acids produces nitrogen, and that nitrogen leaves your body as urea in urine. The second major waste product in urine is creatinine, the breakdown product of creatine phosphate, a molecule stored in muscles and used for rapid energy production. Creatinine is produced at a rate roughly proportional to muscle mass, and unlike urea, it is not significantly reabsorbed or secreted by the tubules. It passes through the kidney almost unchanged. This makes creatinine concentration in the blood a reliable indicator of kidney function. If the kidneys are filtering well, creatinine is cleared from the blood efficiently, and blood levels stay low. If kidney function declines, creatinine accumulates. Serum creatinine is one of the primary blood tests used to assess kidney function, and a rising creatinine is one of the early warning signs of kidney disease. The word urea has an interesting history. Urea was one of the first organic compounds ever synthesized from inorganic materials, and the story of its synthesis matters because it shattered a belief that had held since antiquity. The idea that living things operate by a force, a vital force, a vis vitalis, that is fundamentally different from the forces governing the non-living world. Chemistry that happened in living bodies, according to this view, was special. It couldn't be replicated in a flask. Life was more than chemistry. In 1828, a German chemist named Friedrich Wöhler, working with ammonium cyanate, a thoroughly inorganic salt, accidentally produced urea. He had not been trying to make urea, he had been trying to make ammonium cyanate more pure. Instead, he got crystals that were, chemically, urea, the very compound that kidneys produce from ammonia and excrete in urine, a molecule of life, produced without any life, in a laboratory from a mineral salt. Wöhler wrote to his mentor Jöns Jakob Berzelius with barely contained excitement, "I can no longer, as it were, hold back my chemical urine." The synthesis of urea is often cited as the beginning of organic chemistry as a discipline, and the beginning of the end of vitalism, the belief that living chemistry is categorically different from dead chemistry. Both claims are somewhat exaggerated, but the moment is real. A kidney waste product, produced in a laboratory flask, quietly undermined a philosophical framework that had shaped how people thought about life for 2 millennia. Uric acid is another waste product in urine, the end product of purine metabolism, the breakdown of DNA and RNA. Normally, the kidney filters uric acid and excretes it efficiently. When either production rises or excretion falls, uric acid accumulates in the blood, hyperuricemia, and can crystallize in joints, causing the searing pain of gout. The kidney's ability to handle uric acid is influenced by genetics, diet, alcohol consumption, and medications, which is why gout runs in families, but is also dramatically affected by lifestyle. Then there are the salts: sodium chloride, potassium chloride, calcium, phosphate, magnesium, excreted in amounts precisely calibrated to what you ate minus what you needed to retain. Eat more salt than your body needs, and more sodium appears in the urine. Eat less, and sodium excretion falls. The kidney titrates these outputs continuously, and urine gets its yellow color from urobilin, a pigment derived from the breakdown of bilirubin, which is itself derived from the hemoglobin of old red blood cells. When red blood cells are recycled, which happens continuously, since each red blood cell lives about 120 days, the hemoglobin is broken down into bilirubin, which travels to the liver, is modified, enters the intestine in bile, is further broken down by gut bacteria into urobilinogen, some of which is reabsorbed into the blood and excreted by the kidneys as urobilin. The yellow in your urine is, in a very real sense, the remains of old blood cells. Dehydrated urine is darker because the same amount of urobilin is dissolved in less water. Pale urine means you're well hydrated. Dark yellow or amber means you should drink something. Now let's talk about kidney stones, because approximately 12% of people will have one at some point in their lives, and the experience is memorable enough that most people who've had one remember it vividly and forever. A kidney stone is a solid crystal that forms when the concentration of certain minerals in the urine rises high enough that they begin to crystallize out of solution. The most common type, accounting for about 80% of all kidney stones, is calcium oxalate. Calcium and oxalate are both normally present in urine. The problem arises when they're present in high enough concentrations, and together, that they exceed the solubility limit of calcium oxalate and begin to precipitate. Other types include calcium phosphate, uric acid stones, common in gout, struvite stones, formed in the context of certain bacterial infections, and cystine stones, a rare genetic condition. Each type has its own risk factors, its own prevention strategies, and its own appearance under the microscope. Stones that form in the renal pelvis, the collecting area at the center of the kidney, can remain there asymptomatically for years, growing slowly, causing no pain whatsoever. The pain of kidney stones, the intense pain that sends people to emergency rooms at 2 in the morning, is not from the stone sitting in the kidney. It's from the stone passing through the ureter, the narrow muscular tube that connects the kidney to the bladder. The ureter is only about 3 to 4 millimeters in diameter at its narrowest point. A stone, 2 or 3 millimeters across, can wedge in it, and the ureter, trying to move the stone along with the same peristaltic contractions it uses for urine, contracts against the obstruction. That contraction is visceral pain, mediated by the same nerve fibers that carry intestinal and ovarian pain, and it has a characteristic quality that almost everyone describes the same way: waves. Intense waves, starting in the flank, radiating toward the groin, building to a peak, and then easing slightly, and then building again as the ureter contracts. It is consistently ranked among the most painful experiences in medicine, often compared by people who've experienced both to the pain of childbirth. There is no comfortable position. Moving doesn't help. The pain comes from inside, and there is nothing to be done except wait for it to pass or to have it removed. Small stones, less than 5 millimeters, will usually pass on their own within a few days. But here is something worth understanding about that process. The ureter, at rest, is not a rigid tube. It is a muscular tube, and like the gut, it moves in peristaltic waves, contracting in a coordinated, ring-like pattern that propels urine from the renal pelvis toward the bladder. This peristalsis happens automatically, driven by pacemaker cells in the renal pelvis, about 5 to 8 times per minute. Under normal circumstances, with normal density, freely flowing urine, these contractions accomplish their purpose without sensation. You never feel your ureters moving. The urine arrives in the bladder, and the whole process is transparent. A stone changes everything. When a stone lodges in the ureter, the peristaltic wave cannot propel it. Instead, the wave builds pressure above the obstruction and then collapses, failing. The renal pelvis distends. The pressure inside the kidney rises. Distension of the collecting system, the stretching of the renal pelvis and ureter, is what generates the pain. The pain is not from the stone scraping the ureter, as people sometimes imagine. It is from the distension, and the distension is intermittent, following the rhythm of peristaltic waves that keep arriving and keep failing to move the stone. This is why the pain comes in waves. The wave builds, the ureter contracts, the pressure peaks, the distension is maximal, the pain is worst. Then the wave passes, The pressure temporarily equalizes, and there is a brief easing. Then the next wave comes. The visceral nature of the pain felt deep in the flank, radiating toward the groin and sometimes into the testicle or labia, because the ureter and gonadal organs share nerve supply from the same spinal levels, is one of the reasons kidney stone pain is so disorienting. There is no position of comfort because the pain comes from inside, from an organ you cannot reach and cannot hold. You cannot press against it. You cannot stretch it out. People with kidney stones are famously unable to lie still, pacing, or rocking, which is actually useful clinically. Patients with other causes of severe abdominal pain, like peritonitis, cannot move at all because movement worsens their pain. The kidney stone patient cannot stop moving. It is the kind of pain that overrides everything else and occupies the entire field of awareness. And yet, most stones smaller than 5 millimeters will pass. The ureter gradually dilates around the stone. Hydration helps. More fluid means more urine flow, means more hydraulic force behind the stone. Alpha blockers, the class of drugs used for benign prostatic hyperplasia, also relax the ureter's smooth muscle and can hasten passage. NSAIDs reduce the prostaglandin-mediated inflammation around the stone site and can significantly reduce pain. And eventually, after hours or days that feel considerably longer, the stone reaches the uretrovesical junction, the point where the ureter enters the bladder, which is actually the narrowest point in the urinary tract, and then passes through into the bladder, and from there, out through the urethra, without any significant resistance. The moment of passage is often reported as almost instantaneous relief. The pain that has been consuming everything simply stops. Not gradually, all at once. But here is something worth understanding about that process. The ureter, at rest, is not a rigid tube. It is a muscular tube, and like the gut, it moves in peristaltic waves, contracting in a coordinated, ring-like pattern that propels urine from the renal pelvis toward the bladder. This peristalsis happens automatically, driven by pacemaker cells in the renal pelvis, about 5 to 8 times per minute. Under normal circumstances, with normal density, freely flowing urine, these contractions accomplish their purpose without sensation. You never feel your ureters moving. The urine arrives in the bladder, and the whole process is transparent. A stone changes everything. When a stone lodges in the ureter, the peristaltic wave can't propel it. Instead, the wave builds pressure above the obstruction and then collapses, failing. The renal pelvis distends. The pressure inside the kidney rises. Distension of the collecting system, the stretching of the renal pelvis and ureter, is what generates the pain. The pain is not from the stone scraping the ureter, as people sometimes imagine. It is from the distension, and the distension is intermittent, following the rhythm of peristaltic waves that keep arriving and keep failing to move the stone. This is why the pain comes in waves. The wave builds. The ureter contracts. The pressure peaks. The distension is maximal. The pain is worst. Then the wave passes. The pressure temporarily equalizes, and there is a brief easing. Then the next wave comes. The visceral nature of the pain felt deep in the flank, radiating toward the groin and sometimes into the testicle or labia, because the ureter and gonadal organs share nerve supply from the same spinal levels, is one of the reasons kidney stone pain is so disorienting. There is no position of comfort because the pain comes from inside, from an organ you cannot reach and cannot hold. You cannot press against it, you cannot stretch it out. People with kidney stones are famously unable to lie still, pacing or rocking, which is actually useful clinically. Patients with other causes of severe abdominal pain, like peritonitis, cannot move at all because movement hurts. The kidney stone patient cannot stop moving. It is the kind of pain that overrides everything else and occupies the entire field of awareness. And yet, most stones smaller than 5 millimeters will pass. The ureter gradually dilates around the stone. Hydration helps. More fluid means more urine flow, means more hydraulic force behind the stone. Alpha blockers, the class of drugs used for benign prostatic hyperplasia, also relax the ureter's smooth muscle and can hasten passage. NSAIDs, anti-inflammatory drugs, reduce the prostaglandin-mediated inflammation around the stone site and can significantly reduce pain. And eventually, after hours or days that feel considerably longer, the stone reaches the ureter-vesical junction, the point where the ureter enters the bladder, which is the narrowest point in the urinary tract, and then passes through into the bladder, and from there out through the urethra without any significant resistance. The moment of passage is often reported as almost instantaneous relief. The pain that has been consuming everything simply stops, not gradually, all at once. Larger ones may require medical intervention, shockwave lithotripsy, which uses focused ultrasound from outside the body to break the stone into smaller fragments that can pass, or ureteroscopy, where a thin, flexible scope is passed through the urethra, into the bladder, up the ureter, and the stone is retrieved or fragmented with a laser, or, in extreme cases, surgery. The fact that kidney stones are so common, and that calcium oxalate is the most common type, has led to a great deal of research on dietary factors. Inadequate hydration is the single biggest risk factor. Simply not drinking enough water, so urine is consistently concentrated and calcium oxalate can more easily reach its crystallization threshold. Counterintuitively, reducing calcium intake does not reduce calcium oxalate stone risk. In fact, it may increase it, because dietary calcium binds oxalate in the intestine and prevents it from being absorbed into the blood and ultimately concentrated in the urine. The oxalate content of your diet, spinach, nuts, chocolate, tea, are high in oxalate, matters more than calcium. And sodium intake matters because high sodium intake increases calcium excretion in urine, raising the concentration of the calcium that can combine with oxalate. Drink more water, moderate sodium. Don't restrict calcium. These are the 3 most evidence-backed recommendations for preventing the most common kidney stones. Let's talk about what happens when the kidneys fail. Because chronic kidney disease is one of the most common serious medical conditions in the world, and yet most people with early-stage kidney disease have no symptoms and have no idea they have it. The kidneys have extraordinary functional reserve. You can lose up to about 60% of kidney function, measured by GFR, before any symptoms appear, because the remaining nephrons compensate by working harder, filtering more per unit, doing what they can. This is biologically useful. A single working kidney can sustain life without significant restriction, but medically treacherous, because by the time kidney disease becomes symptomatic, it is often quite advanced. The 2 most common causes of chronic kidney disease in the developed world are diabetes and hypertension. Both make perfect physiological sense given what we know about kidney structure. Diabetes causes chronically elevated blood glucose, and glucose damages the small blood vessels of the glomeruli, the capillaries that do the filtering, leading to a condition called diabetic nephropathy. The glomerular filter becomes leaky, allowing protein that should be retained in the blood to spill into the urine. Over years, the damaged glomeruli scar over and stop working. Hypertension damages the afferent arterioles, the vessels bringing blood to the glomeruli, increasing the pressure inside the glomerular capillaries beyond what the filtration membrane is designed to handle, causing mechanical damage that also scars the glomeruli over time. And kidney disease, as noted, can cause hypertension through the RAS, which then worsens kidney disease, which worsens hypertension, in a cycle that is one of the most frustrating in all of medicine. When kidney function falls low enough, GFR below about 15 milliliters per minute, compared to a normal of 90 to 120, waste products build up in the blood to toxic levels. This is end-stage renal disease, and the syndrome it produces is called uremia, urine in the blood, essentially. Uremia affects virtually every organ system. The brain becomes confused and foggy. The heart develops inflammation of its lining. The lungs fill with fluid that the kidneys can no longer excrete. The skin itches from urea crystals depositing in it. Nausea, loss of appetite, fatigue. Without treatment, it is fatal. The accumulation of uremic toxins, and there are dozens of them, not just urea and creatinine, but a whole array of small molecules that the failing kidney can no longer clear, affects the brain in ways that are distressing to witness and difficult to describe from the inside. The mental cloudiness of uremia is not like ordinary fatigue or ordinary confusion. Patients describe a kind of unreality, a sense that the world is somehow behind glass, that thoughts form slowly and slip away before they can be completed. Concentration becomes effortful and then impossible. Sleep patterns fragment. Personality can change. Irritability, depression, emotional lability. In severe uremia, seizures and coma follow. The body's own metabolic waste, unable to be cleared, accumulates to levels that poison the brain. The skin itching of uremia, pruritus in the clinical language, is one of the most debilitating symptoms and one of the most poorly understood. It is not caused by urea crystals on the skin, as was once believed. The mechanism involves the accumulation of phosphate and other solutes, changes in nerve function, immune activation. It can be constant, intractable, disturbing sleep, destroying quality of life. Patients on dialysis frequently rate pruritus as one of their most burdensome symptoms, sometimes worse than the fatigue, sometimes worse than the dietary restrictions, sometimes worse than the 3 times weekly needle insertions. And the pericarditis of uremia, inflammation of the sac surrounding the heart, caused by uremic toxins, produces a characteristic friction rub that physicians in the pre-dialysis era recognized as a terminal sign. When you heard pericardial friction rub in a patient with kidney disease before 1943, you knew they were dying. The rub was the sound of the end. Dialysis changed what that sound meant. Before the mid-20th century, end-stage kidney disease was a death sentence. There was nothing to be done. Patients with renal failure died, often within weeks of reaching end stage, of fluid overload or hyperkalemia, dangerously high potassium levels that stop the heart, or the accumulation of toxins that no other organ could clear. And then, during the Second World War, in a country under Nazi occupation, a Dutch physician invented a machine that could replace the kidney's filtering function and change the course of medicine. His name was Willem Johan Kolff. He was born in 1911 in Leiden, the Netherlands, and he trained as a physician and became a specialist in internal medicine. When the Nazis occupied the Netherlands in 1940, Kolff moved to a small town called Kampen, partly to get away from the occupation, partly to work somewhere he might have more freedom to pursue his ideas. In Kampen, he watched his first patient die of kidney failure, a 22-year-old man, a farmer, and decided that the death was unacceptable, that there had to be a way to clean the blood artificially. The principle he was working from was simple. If you could pass blood across a semi-permeable membrane, a membrane with pores large enough for small waste molecules to pass through, but small enough to retain the blood cells and proteins, waste molecules would diffuse across the membrane from the blood into a bath of clean fluid on the other side. The blood would be cleaner when it came out than when it went in. Repeat this enough times and you could keep a patient alive. The challenge was doing it in practice in wartime, occupied Netherlands, with no materials, no funding, no support, and constant danger. Kolff used sausage casings, natural cellulose tubing, semi-permeable, already manufactured, as his dialysis membrane. He wrapped the sausage casing around a rotating drum and submerged it in a bath of saline solution. Blood from the patient's artery was pumped into the tubing, traveled around the rotating drum, and the waste molecules diffused out through the casing into the saline bath. The cleaned blood came out the other end and was returned to the patient. His first machine, built in 1943, looked nothing like modern dialysis equipment. It was improvised, held together with ingenuity, and whatever materials he could find in wartime campen. He used an orange juice can for one component. He used parts scavenged from demolished buildings. The rotating drum he built himself, and it didn't work at first. His first 15 patients died, mostly because they arrived too sick, already too far into kidney failure, for even a working machine to save. But he kept refining the machine, and in 1945, his 16th patient, a 67-year-old woman who had been in a uremic coma, survived after a single dialysis session. She lived for another 6 years. Kolff gave away his designs. He sent copies to hospitals in several countries, asking for nothing in return, wanting only for the idea to spread. After the war, he emigrated to the United States, where he continued his work at the Cleveland Clinic and later at the University of Utah, eventually contributing to the development of the artificial heart as well. But the dialysis machine was his first great gift to medicine, built out of sausage casings and an orange juice can in a country under military occupation, given away freely so that other patients would not die the way that 22-year-old farmer had died. Modern hemodialysis is far more sophisticated than Kolff's rotating drum, but the principle is exactly the same. Blood leaves the patient through a needle in a specially created arteriovenous fistula, a surgically joined artery and vein in the forearm that creates a high-flow access point. It passes through tubing into the dialyzer, the artificial kidney, where it flows past a semi-permeable membrane in contact with a specially formulated fluid called dialysate. Waste products diffuse across the membrane. The cleaned blood returns to the patient. The process takes about 3 to 4 hours and must be done 3 times a week for the rest of the patient's life, unless they receive a transplant. The transplant option was developed in parallel with dialysis, and it required solving a different problem entirely. Not mechanical filtration, but biological compatibility. You can give someone a new kidney, but the immune system, designed to reject anything foreign, will attack and destroy it unless that rejection is prevented. This is the fundamental challenge of transplantation, and solving it, partially, imperfectly, but sufficiently, is one of the great stories of 20th century medicine. The first successful kidney transplant was performed on December 23, 1954, at Peter Bent Brigham Hospital in Boston. The surgeon was Joseph E. Murray, who would later receive the Nobel Prize in Physiology or Medicine in 1990 for the work. The patient was a 23-year-old man named Richard Herrick, who was dying of chronic nephritis and end-stage kidney disease. His identical twin brother, Ronald, agreed to donate one of his kidneys. Murray chose identical twins because they are genetically identical and therefore immunologically identical. The immune system of one would not recognize the kidney of the other as foreign. There was no immunosuppression to speak of. They were counting on the genetic match to prevent rejection, and it worked. Richard Herrick recovered, lived for 8 years after the transplant, long enough to marry his nurse and have 2 children, before dying of unrelated kidney disease in his remaining native kidney. Ronald Herrick lived until 2010, donating a kidney having made no significant difference to his own health. The transplant was, by any measure, a triumph. But the triumph of identical twin transplantation raised an obvious next question. Could transplantation work between people who were not identical twins? Between people whose immune systems were different? This required solving the rejection problem, and that required immunosuppression, drugs that dampen the immune system enough to prevent it from destroying the new kidney. The first breakthrough came with azathioprine, developed in the early 1960s by George Hitchings and Gertrude Elion, who would also win the Nobel Prize in 1988 for their work on drug development. Azathioprine could suppress the immune system enough to allow transplants between non-identical individuals, though with significant toxicity and variable success. The real transformation came in the 1970s with the discovery of cyclosporine, a drug derived from a Norwegian soil fungus that selectively suppresses the T cells responsible for graft rejection without broadly shutting down all immune function. Cyclosporine transformed kidney transplantation from an experimental procedure with modest success rates into a routine operation with excellent outcomes. Today, kidney transplantation is the preferred treatment for end-stage kidney disease. Better outcomes than dialysis, better quality of life, longer survival. But demand vastly exceeds supply. In the United States alone, about 100,000 people are on the waiting list for a kidney at any given time, and about 13 people die every day waiting for one that doesn't come in time. The biology has been substantially solved. The ethics and the logistics remain deeply challenging. Let's step back from the dramatic and return to the quiet, because while the history of dialysis and transplantation is remarkable, what I want you to think about right now is the much quieter thing that has been happening throughout this entire conversation: your own kidneys, working. Think about what they're doing right now. While you've been lying here, your GFR has adjusted slightly for your horizontal position and your decreased activity. Blood flow to the kidneys changes with posture, and the kidney accommodates. Your ADH levels have been rising steadily as you've moved toward sleep, not because you're dehydrated, but because vasopressin follows a circadian rhythm with higher levels at night, which causes the kidney to concentrate urine during the sleeping hours. This is why you don't need to urinate as often at night as during the day. Your kidneys are producing less urine right now than they will after breakfast, not because they're resting, but because they've shifted into a conservation mode, calibrated to the fact that you're unlikely to be drinking during the night. The kidney has its own circadian clock. It's one of the organs that most strongly expresses circadian variation in its function. Filtration rates, sodium handling, acid-base regulation, renin release, all vary with time of day in ways that have been only partially mapped. The kidney knows what time it is, in a sense, and behaves differently at 3 in the morning than at 3 in the afternoon. There are clock genes expressed in kidney cells, the same molecular clockwork present in the brain's suprachiasmatic nucleus and in the liver and in most organs in the body. Disrupting these rhythms through shift work, through jet lag, through chronic sleep disruption, disrupts kidney function in ways that researchers are still trying to quantify. There is emerging evidence linking circadian disruption to increased kidney stone risk, to higher rates of proteinuria, to faster progression of chronic kidney disease. The kidney, like everything else, functions best with regular sleep. Right now, the tubular cells along the length of your nephrons are operating at different rates than they will tomorrow afternoon. The transporters in the proximal tubule, the ones that haul glucose and amino acids and sodium out of the filtrate and back into the blood, have a slightly different activity profile at night. The cells of the collecting duct respond to ADH a little differently as dawn approaches. The juxtaglomerular cells that produce renin are calibrated to the time. The kidney is not a static machine running at a fixed speed. It is a dynamic, temporally organized system embedded in the rhythms of the body as a whole. And your blood right now, at this moment, is being maintained at a specific pH, a specific sodium concentration, a specific osmolarity, a specific volume. All of those parameters are within tight ranges because two organs the size of your fists, sitting quietly behind your abdominal cavity, are continuously adjusting the composition of the fluid that circulates through every cell in your body. The chemistry of your brain right now, the ions that cross the membranes of your neurons as you listen to these words, as your thoughts slow and soften toward sleep, depends on the kidney's work. The electrical potential of your heart cells, the signaling chemistry of every organ, the stability of your blood pressure, all of it is downstream of what your kidneys are doing. Let's talk for a moment about dialysis from the patient's perspective, because most people have a vague impression of it as unpleasant without really knowing why. The experience of being on dialysis is one of the most medically demanding things a person can go through while remaining technically an outpatient. A dialysis patient, assuming hemodialysis 3 times a week, which is by far the most common form in the United States and most of the developed world, typically spends 12 to 15 hours per week connected to a machine. The needle insertions into the fistula are not trivial. The fistula itself, the surgically created arteriovenous connection in the forearm, creates a distinctive thrill, a palpable buzz under the skin, and over years of use, the vessels can dilate into structures visible and palpable from across the room. A blood flow of 300 to 500 milliliters per minute passes through the dialyzer for hours. Most patients experience a drop in blood pressure during dialysis, sometimes severe enough to cause cramping, nausea, or loss of consciousness. Fatigue after a session is profound and often lasts hours. Between sessions, patients must adhere to strict dietary restrictions. Potassium must be limited because the kidneys can no longer excrete excess potassium, and hyperkalemia can stop the heart. Phosphorus must be limited because kidney disease impairs phosphorus excretion, and high phosphorus levels damage blood vessels and bones. Fluid must be limited because the kidneys can no longer excrete excess water, and patients on dialysis rely on the 3 times weekly sessions to remove fluid that has accumulated. A patient on hemodialysis who drinks too much fluid between sessions may arrive for dialysis several kilograms heavier, with the excess entirely as fluid that has distributed into their lungs and ankles and belly. Some patients do peritoneal dialysis instead of hemodialysis. In peritoneal dialysis, the filtering membrane is not an artificial membrane in a machine, but the patient's own peritoneum, the thin lining of the abdominal cavity, which is highly vascular and semi-permeable. A tube is surgically inserted into the abdomen, and several times a day, the patient fills the abdominal cavity with 1 liter or 2 of dialysate fluid, lets it dwell for several hours, while waste products diffuse across the peritoneum from the blood into the fluid, and then drains it out and replaces it. It can be done at home, at night, with a cycling machine that automates the exchanges during sleep. It's gentler than hemodialysis, less cardiovascular stress, no needles, but it works less efficiently for large patients, and the long-term risk of peritoneal membrane failure after years of use is a real limitation. There is a third approach to renal replacement therapy that is less common in the United States but important to know about: nocturnal home hemodialysis. Some patients, particularly those who do their own dialysis at home, dialyze more frequently, 5 or 6 nights a week, for 6 to 8 hours each session while they sleep. The outcomes from nocturnal dialysis are substantially better than conventional 3 times weekly dialysis. Better blood pressure control, better phosphate management, better survival. The reason is simple. More time on dialysis means more toxin removal and better fluid control. Conventional 3 times weekly dialysis was invented in the context of limited machine availability and the need for patients to travel to dialysis centers. It's a logistical schedule, not necessarily an ideal medical one. Patients who can do it at home while sleeping and who choose to do it more frequently do better by most measures. But it requires motivation, technical training, a suitable home environment, and support infrastructure, and only a minority of dialysis patients pursue it. There is also the option of conservative management, choosing not to pursue dialysis at all in patients who are very elderly or very ill, where dialysis might extend life by months, but at significant cost to quality of life. This is an ethically complex conversation that nephrologists have with some patients, particularly older adults with many other serious medical conditions. Dialysis in a 90-year-old with heart failure and dementia may offer 3 to 6 months of life extension, but those months will include 3 sessions a week of a demanding and uncomfortable procedure, strict dietary restriction, frequent hospitalizations. Some patients, when the full picture is explained, choose to forego dialysis and focus on comfort and quality of life. Renal palliative care is an emerging subspecialty. The kidney's failure in the very elderly and very ill sometimes raises questions that go well beyond physiology. Neither form of dialysis replaces the full function of a kidney. Both can clear small molecules reasonably well. Neither can produce EPO, activate vitamin D, control blood pressure through the RAS with the full subtlety the kidney uses. Neither can respond dynamically to what the body is doing moment to moment, exercising, sleeping, eating a salty meal, sweating, the way a living kidney does. Dialysis patients have higher rates of cardiovascular disease, higher rates of infection, significantly shortened lifespans compared to the general population, and often a persistent sense of fatigue and malaise that reflects the difference between continuous, dynamic kidney function and 3 sessions a week of passive diffusion. This is not to diminish what dialysis offers. It offers life, where otherwise there would be none. The approximately 500,000 Americans on dialysis at any given time are alive because of a machine invented from sausage casings in wartime Netherlands. The treatment is remarkable, but the kidney is more remarkable still, and it's worth understanding how much the machine doesn't do in order to appreciate what the organ does. Let's spend some time with the kidney's role in regulating not just waste but water, because water regulation, the precise control of how much water is in your body, where it is, and what concentration of solutes it contains, is arguably the kidney's most important job, and it's a job that has profound connections to every aspect of health. The human body is about 60% water by weight. That water is distributed across multiple compartments, inside cells, outside cells, in the interstitial fluid, and in the blood plasma. The volumes and osmolalities of these compartments are carefully regulated, and the kidney is the primary regulator. It does this through the integrated actions of ADH, aldosterone, atrial natriuretic peptide, a hormone produced by the heart when it's overstretched by excess fluid, which acts on the kidney to increase sodium and water excretion, and the intrinsic autoregulatory mechanisms of the nephron itself. When you drink a liter of water rapidly, your blood osmolality falls slightly, diluted by the incoming water. This drop is detected within minutes by osmoreceptors in the hypothalamus, the part of the brain that monitors blood composition. The hypothalamus signals the posterior pituitary to reduce ADH secretion. ADH levels in the blood fall. The collecting duct, deprived of ADH, becomes impermeable to water. The dilute filtrate flows through without losing its water, and you produce large volumes of dilute urine, sometimes as much as 15 to 20 milliliters per minute, compared to a baseline of about 1 milliliter per minute. Within an hour or two, the excess water has been excreted, blood osmolality has returned to normal, and ADH levels have risen back to their resting level. The system has corrected itself, quickly and precisely, without any conscious awareness on your part. When you sweat heavily and don't drink enough, during exercise, in heat, the reverse happens. Blood volume falls, blood osmolality rises, the hypothalamus detects both changes, ADH secretion rises, the collecting duct becomes highly permeable to water, urine becomes maximally concentrated, the kidneys hold on to every drop they can. You feel thirsty, because the same hypothalamic neurons that control ADH also drive thirst behavior. The system is pushing you both to conserve what you have and to take in more. This dynamic system is why dehydration and overhydration are both potentially dangerous. Severe dehydration, losing 10% or more of body water, leads to confusion, rapid heart rate, falling blood pressure, and ultimately cardiovascular collapse. But severe overhydration, drinking extreme quantities of plain water, as happens sometimes in endurance events or in certain psychiatric conditions, can lead to hyponatremia, dangerously low blood sodium, as the blood is diluted faster than the kidneys can excrete the excess water. Hyponatremia can cause brain swelling, seizures, coma, and death. The kidney can only excrete water so fast, overwhelm it, and the system fails. For most people, most of the time, the kidney handles these challenges effortlessly, but the effortlessness is an illusion of competence. It looks easy because the system is so well designed. The nephron is processing filtrate continuously, adjusting at every step, responding to hormonal signals that are themselves responding to blood composition measurements taken in real time. It looks like nothing is happening. A lot is happening. Now, one of the aspects of kidney physiology that most people never encounter is the concept of tubuloglomerular feedback, the way the kidney regulates its own filtration rate from within the nephron itself. At the point where the ascending limb of the loop of Henle reaches the cortex and begins the distal convoluted tubule, there is a specialized region called the macula densa, a small cluster of cells whose name means dense spot in Latin because they are packed tightly together and look distinct under the microscope. The macula densa cells are tasting the filtrate, sensing its sodium chloride concentration, and they are doing this, critically, at a point that sits anatomically right next to the afferent arteriole of the same nephron, right next to the juxtaglomerular cells. If the concentration of sodium chloride at the macula densa is too high, meaning too much filtrate is being produced, more than the tubule can efficiently process, the macula densa sends a signal to the afferent arteriole that causes it to constrict, reducing blood flow into the glomerulus, reducing filtration pressure, reducing the rate at which filtrate is produced. If sodium chloride concentration at the macula densa is too low, meaning filtration is inadequate, the afferent arteriole relaxes, blood flow increases, and filtration rate rises. Each nephron is thus monitoring its own output and adjusting its own input. 1 million of these feedback loops are running simultaneously in each kidney, in parallel, each one independently calibrating its filtration rate based on what is arriving at its own macula densa. The aggregate behavior of all these independent loops is the remarkably stable GFR that healthy kidneys maintain. It is a distributed control system, decentralized, with no central command, just a million local feedback loops, each one doing its small part of the overall regulation. This architecture has a consequence that's clinically important. It means GFR is relatively stable even when blood pressure varies considerably, because the tubuloglomerular feedback is always working to compensate. If blood pressure rises, filtration would tend to increase, but tubuloglomerular feedback senses the increase and constricts the afferent arteriole to bring filtration back to normal. If blood pressure falls, filtration would tend to decrease, but tubuloglomerular feedback senses the decrease and dilates the afferent arteriole to maintain filtration. This autoregulation keeps GFR stable across a blood pressure range of about 70 to 170 millimeters of mercury, a large range that covers most of the variation a person experiences in daily life. Outside that range, the autoregulation fails and GFR becomes pressure-dependent. Extreme hypotension, as in shock, overwhelms the system and filtration essentially stops. This is acute kidney injury in its most acute form. The kidney, deprived of blood flow, simply cannot filter. It's worth saying something about the kidney and pregnancy, because pregnancy creates a remarkable set of demands on renal physiology that most people never consider. During pregnancy, cardiac output rises dramatically. The heart pumps substantially more blood per minute to supply both the mother and the developing placenta. Renal blood flow increases in parallel, and GFR rises by roughly 50% above baseline by the end of the first trimester. A GFR that would indicate mild kidney disease in a non-pregnant person might be normal in a pregnant one. Conversely, a GFR that seems normal in a pregnant person might actually represent significant underlying disease. The kidney must also accommodate the massive changes in blood volume during pregnancy. Blood volume increases by 30 to 50%. Progesterone relaxes smooth muscle, which causes the ureters to dilate and become less effective at moving urine from the kidney to the bladder, a condition called physiological hydronephrosis of pregnancy, which predisposes pregnant women to urinary tract infections and kidney infections. The renin-angiotensin system is dramatically activated during normal pregnancy, yet blood pressure typically falls rather than rises, because the vasodilatory effects of other pregnancy hormones counteract the vasoconstrictive effects of angiotensin II. When this balance fails, when the vasodilation is insufficient, or when the renin-angiotensin system becomes pathologically overactive, preeclampsia results. High blood pressure, protein in the urine, dangerous complications for both mother and fetus. Preeclampsia is fundamentally a kidney disease. The glomeruli become damaged and leaky, proteinuria develops, and the entire renal regulation of blood pressure becomes dysregulated. It affects 5 to 8% of pregnancies and is one of the leading causes of maternal and fetal morbidity worldwide. Understanding preeclampsia required understanding kidney physiology and developing treatments for it, careful control of blood pressure, magnesium sulfate to prevent seizures, delivery of the baby as the definitive treatment, required understanding what the kidney was doing and why. The kidney is implicated in one of the most dangerous complications of one of the most common human experiences. Let's think about the kidney in the context of evolution, because understanding why the kidney is designed the way it is requires understanding the evolutionary pressures that shaped it. The basic challenge of water and salt regulation is ancient. It appears in simple multicellular organisms and has been a central problem in biology since animals first left the sea. Ocean fish, living in a medium of essentially the same salt concentration as their body fluids, have relatively simple kidneys. They don't need to regulate osmolality much because their environment does it for them. Freshwater fish face the opposite problem. Their body fluids are saltier than the water around them, so they constantly absorb water by osmosis and must produce enormous volumes of very dilute urine to get rid of it. Marine fish do the opposite. Their body fluids are less salty than seawater, so they constantly lose water to their environment and must conserve every drop. When vertebrates moved onto land, the challenge of water conservation became paramount. There was no surrounding medium to buffer osmolality. The body had to contain its own ocean, carry its own salt bath, and maintain it without constant exchange with the environment. The mammalian kidney, with its remarkably efficient concentration mechanism, the loop of Henle, the collecting duct's ADH response, the full elaborate system, is the solution to the problem of living in air. It is, in a sense, the technology that made land life possible for vertebrates. The desert rodent, the kangaroo rat of the American Southwest, the jerboa of the Sahara, can survive its entire life without drinking, extracting enough water from its food and from the metabolic water produced by burning carbohydrates and fats, and conserving every drop through kidneys that can produce urine concentrated to 9,000 milliosmoles per kilogram, nearly 8 times what a human kidney can manage. The kangaroo rat has a loop of Henle proportionally longer than any other mammal, generating a deeper concentration gradient in its medulla, producing correspondingly drier urine. The loop of Henle is the evolutionary dial for desert adaptation, and it has been turned as far as it goes in animals that live where water is most scarce. The beaver is an interesting intermediate case. Beavers are semi-aquatic rodents living in freshwater environments, and their kidneys reflect this. Moderate concentrating ability, neither as impressive as a kangaroo rat's, nor as limited as a fully aquatic animal's. Marine mammals face a completely different challenge than freshwater ones. A dolphin living in saltwater cannot drink the sea around it without making things worse. Seawater is more concentrated than mammalian body fluids, so drinking it would cause a net loss of water through osmosis. Marine mammals get their fresh water primarily from the fish and squid they eat, which contain adequate amounts of fresh water, and their kidneys, while not dramatically superior to human kidneys in concentrating ability, are efficient enough to handle the modest salt loads and produce adequately concentrated urine from their metabolic water budget. Whales and dolphins that strand on beaches and spend time in the sun sometimes become dehydrated in the way desert animals do, not from drinking, but from radiating heat into dry air, which underscores how carefully balanced the marine mammal water economy actually is. The bird kidney is a slightly different design from the mammalian kidney. Birds have 2 kinds of nephrons, mammalian type ones with loops of Henle and more reptilian type ones without, and this hybrid design reflects their evolutionary history and the demands of flight. Concentrated urine requires loop of Henle function. A kidney with fewer loops produces less concentrated urine. The trade-off for birds is that the kidney can be made lighter by reducing loop abundance. Seabirds that drink salt water have additionally evolved salt glands, organs near the nostrils that can excrete extremely concentrated salt solution, bypassing the kidney entirely for sodium chloride handling. It's a different engineering solution to the same osmoregulatory problem, arrived at independently. The beaver is an interesting intermediate case. Beavers are semi-aquatic rodents living in freshwater environments, and their kidneys reflect this. Moderate concentrating ability, neither as impressive as a kangaroo rat's, nor as limited as a fully aquatic animal's. Marine mammals face a completely different challenge. A dolphin living in saltwater cannot drink the sea around it without making things worse. Seawater is more concentrated than mammalian body fluids, so drinking it would cause a net loss of water through osmosis. Marine mammals get their fresh water primarily from the fish and squid they eat, and their kidneys, while not dramatically superior to human kidneys in concentrating ability, are efficient enough to handle modest salt loads and produce adequately concentrated urine. Seabirds that drink salt water have additionally evolved salt glands near the nostrils that can excrete extremely concentrated salt solution, bypassing the kidney entirely for sodium chloride handling. It is a different engineering solution to the same osmoregulatory problem, arrived at independently, through an entirely different evolutionary pathway. The problem: how do you keep your internal ocean stable when the external world has different chemistry, is ancient and universal. The solutions are varied and fascinating, and in every case beautiful in their specificity. Humans are not desert specialists by this measure. Our maximum urine concentration of about 1200 milliosmoles is modest. We evolved near water, in environments where drinking was possible. But we carry the mechanism, and it works, and right now it is calibrated to the fact that you drank some amount of water today, in an environment where water was available, and you'll sleep through the night without drinking, and by morning your urine will be more concentrated than it was at noon. The mechanism is ancient and shared with every mammal. Your kidneys, in their essential architecture, would be recognizable to a paleontologist studying an early mammal fossil from the Triassic. Now, let's come back to the present, and to you, to the specific situation of lying here, in the dark, breathing slowly. Your kidneys are operating in their nocturnal mode. ADH is elevated. The pituitary has been releasing more of it as you've moved toward sleep, because the circadian clock that governs ADH secretion is tied to the sleep-wake cycle. Your urine output is lower than it was this afternoon. Your urine is more concentrated, and you will almost certainly not need to urinate until morning, unless you have a condition called nocturia, from the Latin nox, night, and urina, urine, where the normal nocturnal suppression of urine output fails. There is a related phenomenon worth knowing about: the first morning urine. The urine you produce first thing in the morning, after a night of elevated ADH and minimal fluid intake, is the most concentrated your kidneys can make. It is in this sample that many diagnostic markers are most reliably detected, because they are most concentrated. Protein in the urine, if present, is easiest to detect in the first morning sample. Human chorionic gonadotropin, the hormone detected by pregnancy tests, is most reliably present at high concentrations in first morning urine, which is why pregnancy tests work most reliably on that sample. Glucose, if spilling into the urine in diabetes, will be detectable all day. But the first morning sample gives the clearest picture. The first morning urine is, in a sense, the kidney's daily report, the summary of its overnight work, concentrated and informative. There is a related phenomenon worth knowing about: the first morning urine. The urine you produce first thing in the morning, after a night of elevated ADH and minimal fluid intake, is the most concentrated your kidneys can make. It is in this sample that many diagnostic markers are most reliably detected. Protein in the urine, if present, is easiest to detect in the first morning sample. Human chorionic gonadotropin, the hormone detected by pregnancy tests, is most reliably present at high concentrations in first morning urine, which is why pregnancy tests work most reliably on that sample. The first morning urine is the kidney's daily report, the summary of its overnight work, concentrated and informative. And the dark yellow of that first glass is not cause for alarm. It is your kidneys, having done their job through the night, handing back the evidence. Nocturia is actually quite common, affecting perhaps 30% of adults over the age of 30, and it disrupts sleep significantly. Waking up to urinate once a night is classified as nocturia. Waking twice or more is more severe and is associated with substantially higher rates of sleep disruption, fatigue, and, in older adults, particularly, falls. The causes are many. poorly controlled diabetes. More glucose in the blood means more in the filtrate, and osmotic diuresis, water following the glucose, means more urine. Heart failure, fluid that pools in the legs during the day, redistributes when you lie down, increasing venous return to the heart and triggering atrial natriuretic peptide release, which tells the kidneys to produce more urine. Obstructive sleep apnea, the effort of breathing against an obstructed airway, generates negative intrathoracic pressure, which is perceived by the heart as high pressure, which also triggers natriuretic peptide release. Benign prostatic hyperplasia in men. The enlarged prostate obstructs the bladder outlet, and the bladder never fully empties, so it fills and triggers urgency faster, and simply age-related decline in the kidney's circadian variation, so that the nocturnal suppression of urine output becomes less robust. None of these things are happening to you, presumably, or you'd know about it. For you, right now, the normal nocturnal program is running. ADH is high. Your nephrons are reabsorbing water from the collecting duct with the quiet efficiency they always do. Your blood pressure is slightly lower than it was this afternoon. Blood pressure dips during sleep, a phenomenon called nocturnal dipping, and the kidneys accommodate it. Your heart rate has slowed. Your metabolic rate has dropped. Your kidneys have adjusted their filtration rate accordingly. And somewhere in the medulla of each kidney, in the inner reaches of the tissue where the concentration gradient is steepest, where the interstitial osmolarity reaches its maximum of 1,200 milliosmoles per kilogram, there, in those cells, the water is being drawn out of the collecting duct by the force of that gradient, pulled through channels called aquaporins, tiny protein pores in the cell membrane that are specifically permeable to water molecules, and nothing else. Aquaporins were discovered in 1992 by Peter Agre, who shared the Nobel Prize in Chemistry in 2003 for the discovery. There are 13 types of aquaporins in the human body. The kidney uses several of them, with different types in different segments of the tubule, each one doing its specific part of the water transport job. The aquaporins in the collecting duct are regulated by ADH. When ADH binds to its receptor on collecting duct cells, it triggers a cascade that inserts aquaporin channels into the cell membrane, making the membrane permeable to water. When ADH levels fall, the aquaporins are retrieved from the membrane and permeability drops. This molecular level control, a hormone binding to a receptor, triggering a signaling cascade, causing specific protein channels to insert into a membrane, thereby changing the water permeability of an entire tubular segment, is the mechanism underlying your body's response to dehydration. It is happening right now, in billions of cells distributed across the medulla of your kidneys, controlled by a hormone whose concentration in your blood has been rising for the past hour as you've approached sleep, the molecular machinery of homeostasis, running while you rest. There's something I want you to sit with for a moment. We've talked about filtration, about the glomerulus and Bowman's capsule, about the loop of Henle and its elegant countercurrent multiplication. We've talked about hormones, ADH, aldosterone, renin, EPO, calcitriol. We've talked about acid-base balance and urine composition and kidney stones, and dialysis and transplantation. All of that is true, and all of it is happening, in some measure, right now in your body. But I want to step back from the mechanism and just say, the thing that the kidneys are doing at the highest level of description is maintaining the conditions for life. Every cell in your body, every neuron, every muscle cell, every immune cell, every red blood cell, lives in a fluid environment. The composition of that fluid, its salinity, its pH, its osmolarity, its levels of potassium and calcium and phosphate, must remain within narrow bounds for those cells to function. Neurons can't fire at the wrong pH. Muscle cells can't contract if potassium is too high. Red blood cells can't carry oxygen if the blood is too dilute or too concentrated. The internal environment has to be right, and maintaining it right, across everything you eat, everything you drink, everything you sweat out, everything your metabolism produces, is the kidney's job. It is the continuous, unending, automatic work of maintaining the chemical ocean that all your cells live in. This is what the French physiologist Claude Bernard meant when he coined the phrase milieu intérieur, the internal environment, in the 19th century. And it is what Walter Cannon meant when he coined the word homeostasis in 1926, the tendency of living systems to maintain stability. The kidney is the primary organ of homeostasis, more than the liver, more than the lungs, more than any other organ except possibly the brain. The kidney is responsible for the stability of the internal environment that makes everything else possible. You never feel this happening. You don't sense your blood osmolality. You don't sense your GFR. You don't sense the renin your juxtaglomerular cells are releasing, or the aldosterone your adrenal glands are producing in response. The system runs entirely below the level of consciousness. It has been running since before you were born. The fetal kidneys begin producing urine by about 10 weeks of gestation, and while the placenta takes over most homeostatic regulation during fetal life, the kidneys are practicing, getting ready for the moment when they have to do it on their own. The moment you were born and the placenta was removed, your kidneys took over, and they've been doing it every minute since. We're getting close to the end, but I want to spend a moment with one more thing, the question of what kidney disease actually feels like in the early stages. Because there is something medically important and in its way philosophically interesting about an organ whose loss is invisible until the very late stages. Chronic kidney disease is often called a silent disease, and the silence is almost total through stages 1, 2, and 3, which correspond to GFR values above 30 milliliters per minute. At these stages, waste products are still being cleared adequately. Blood pressure may be only mildly elevated. There may be some protein in the urine, detectable with a simple dipstick test at any doctor's office, but no symptoms whatsoever. The person feels fine. They may have had a blood test for other reasons and noticed that their creatinine is slightly elevated, without knowing what that means. They may be told they have early kidney disease and feel no different than they did the day before. The loss of kidney function is slow, usually, and the body's compensation, the remaining nephrons working harder, masks it. There is no pain. There is no fatigue, not yet. There is nothing to point to and say, there, that is where the problem is. The disease advances, if it does, invisibly. By the time symptoms appear, the fatigue, the edema, the difficulty concentrating, the nausea, much of the kidney's reserve is gone. This is one of the strongest arguments for routine screening in high-risk populations. People with diabetes, people with hypertension, people with a family history of kidney disease, people who are older. A simple blood creatinine or a urine test for protein can catch chronic kidney disease when it's in stages where progression can be slowed, where blood pressure control, diabetes management, reduction of protein in the diet, and avoidance of nephrotoxic medications can meaningfully delay or even prevent end-stage disease. The kidneys can be protected if they're found to be at risk, but they can't announce themselves as at risk. They don't know how to do that. They just quietly continue working, compensating, adjusting, filtering, until they can't anymore. There is a model of the kidney I want to leave you with. Not a technical model, not a diagram, but a way of thinking about it. Let's think, for a moment, about the kidney in the context of what it means for a living organism to persist through time. You are not the same collection of molecules you were 7 years ago. The atoms in your body are constantly being replaced. through food, through breath, through the normal turnover of cellular components. The proteins in your muscle cells are broken down and rebuilt. The lipids in your cell membranes are replaced. Even the calcium in your bones, though it turns over slowly, is not the same calcium it was a decade ago. You are a pattern, not a fixed collection of stuff, a form that persists through continuous material exchange with the world. The kidney is what makes this persistence possible. Without the continuous clearing of waste products from that material exchange, the byproducts of metabolism would accumulate to toxic levels. Without the continuous regulation of fluid composition, the internal environment that all your cells depend on would drift away from the narrow range in which they can function. You persist not despite the constant exchange, but because of the constant clearing. The kidney is, in this sense, one of the organs most responsible for the fact that you are still you, still recognizably yourself, still a coherent functioning system, from one day to the next. The philosopher Derek Parfit wrote extensively about personal identity, about what makes you the same person today as the person who was born, who was 5 years old, who will grow old, in the same body. His answer, roughly, was that identity is a matter of psychological continuity, of overlapping connections of memory and intention and experience. That's probably right at the psychological level, but at the biological level, the answer involves the kidney. What makes you continuous is the maintenance of a stable internal environment. The same blood chemistry, the same cellular operating conditions, the same milieu intérieur that allows your brain to go on running the same neural patterns, accumulating the same memories, pursuing the same intentions. The psychological continuity is downstream of the biological stability, and the biological stability is, to a significant degree, downstream of the kidney. This is an unusual way to think about an organ, but it is, I think, a correct one. The kidney is not just a filter, it is part of the infrastructure of selfhood. Let's think, for a moment, about what it means for a living organism to persist through time. You are not the same collection of molecules you were 7 years ago. The atoms in your body are constantly being replaced, through food, through breath, through the normal turnover of cellular components. The proteins in your muscle cells are broken down and rebuilt. The lipids in your cell membranes are replaced. Even the calcium in your bones, though it turns over slowly, is not the same calcium it was a decade ago. You are a pattern, not a fixed collection of stuff, a form that persists through continuous material exchange with the world. The kidney is what makes this persistence possible. Without the continuous clearing of waste products from that material exchange, the byproducts of metabolism would accumulate to toxic levels. Without the continuous regulation of fluid composition, the internal environment that all your cells depend on would drift away from the narrow range in which they can function. You persist, not despite the constant exchange, but because of the constant clearing. The kidney is, in this sense, one of the organs most responsible for the fact that you are still you, still recognizably yourself, still a coherent functioning system, from one day to the next. The philosopher Derek Parfit wrote extensively about personal identity, about what makes you the same person today as the person who was 5 years old, who will grow old. His answer, roughly, was that identity is a matter of psychological continuity, overlapping connections of memory and intention and experience. That is probably right at the psychological level, but at the biological level, the answer involves the kidney. What makes you continuous is the maintenance of a stable internal environment, the same blood chemistry, the same cellular operating conditions, the same milieu intérieur that allows your brain to go on running its neural patterns, accumulating memories, sustaining intentions. The psychological continuity is downstream of the biological stability, and the biological stability is, to a significant degree, downstream of the kidney. This is an unusual way to think about an organ, but it is a correct one. Your kidney is a correspondence system. Every moment, it is reading the state of your body, the composition of your blood, its pressure, its volume, its pH, its oxygen content, its hormone levels, and it is responding with adjustments. It is producing or withholding hormones. It is tightening or loosening the filtration rate. It is holding more water or releasing more water. It is shifting the acid-base balance one way or another. It is telling the bone marrow to make more red blood cells or telling it to ease off. It is activating vitamin D or not. It is executing 100 simultaneous conversations with the rest of your physiology, all of them ongoing, all of them dynamic, all of them aimed at the same goal, keeping the internal environment stable. And it does all of this in a structure the size of your fist, in 1 million tiny nephrons, each one working independently, each one contributing its small adjustments to the aggregate outcome, in a tissue that weighs about 150 grams per kidney, in an organ that, if you saw it on a plate, you would think was too small to be responsible for so much. Kolff saw a young man die of kidney failure in 1940 and decided it was unacceptable. He was right. It was unacceptable. And it was unacceptable not because kidney failure is dramatic. It often isn't. It's quiet and slow. But because what the kidney does, when it's working, is so fundamental and so constant and so vast that losing it cannot be survived without help. You need the internal ocean to stay balanced. You need the waste to be cleared. You need the blood pressure to be regulated and the red blood cells to be stimulated and the vitamin D to be activated and the acid to be excreted. Without the kidney, all of those systems start to drift. The internal environment destabilizes. The conditions for life erode. But the kidney is working. Tonight, for most people reading or listening to this, both kidneys are working. They filtered your blood 60 or 70 times today. They recovered the glucose and the amino acids and the bicarbonate and the water. They excreted the urea and the creatinine and the excess salt and the metabolic acids. They maintained your blood pressure and produced EPO and activated vitamin D and regulated your osmolality and kept your pH in the 7.35 to 7.45 range it has been in every day of your life. They did all of this without asking anything of you, without your knowledge, without your attention, without your awareness. They did it while you were sleeping, and while you were eating, and while you were exercising, and while you were doing whatever you did today. They did it last night, and the night before, and every night going back to the night you were born. They will do it tomorrow night, and the night after that. So here's what I want you to think about as you drift off, if you haven't already. Somewhere below your ribs right now, 2 fist-sized organs are doing the most quietly extraordinary work in your body. 180 liters of filtrate will cycle through them by tomorrow morning. The composition of the fluid around every cell in your body will remain, as it has always remained, exactly where it needs to be. Your body's internal ocean will stay balanced. The conditions for your life, for your sleep, for your dreams, will be maintained, as they always are, as they always have been, by organs you have almost certainly never had cause to think about tonight. Your kidneys are working. You don't have to do anything. You just get to rest. Rest well.